Over the past week, we've been going over one of the major metabolic pathways that occurs in AP biology, cellular respiration. Today, we're going to switch gears and look at the other major metabolic pathway that's covered, photosynthesis. So first off, let's look at the chemical formulas for each of these. Cellular respiration, as we've already discussed, involves taking a glucose molecule, taking oxygen, and a series of chemical reactions is going to produce carbon dioxide, water, and energy. And in particular, the energy is in the form of ATP, which is a form that cells are able to use at a moment's notice. Let's contrast this to photosynthesis. If you take a look at photosynthesis, what you'll see is our reactants are carbon dioxide and water. It's powered by sunlight energy. And at the end, you end up with C6H12O6, glucose, and oxygen. If you take a look carefully at these two chemical reactions, you'll realize they're opposites of each other. They're virtually the same thing if you were to reverse the arrow. The difference between them, however, is what occurs during the arrow, and they each have a different function. Cellular respiration is designed to release the energy captured in a glucose molecule and turn it into ATP so our cells can use it. Photosynthesis is designed around the idea of taking energy from sunlight and capturing that energy and placing it in a glucose molecule for long-term storage until we need to use it again. Before we go any further, I want to clarify a very, very common mistake that many people have about respiration and photosynthesis. And to do that, I need to introduce two different terms to you. The first term is autotroph. And the second is heterotroph. Autotroph and heterotroph are two categories of living things, organisms, divided based on how they get their food. Autotrophs are organisms that produce their own food using photosynthesis. Heterotrophs, on the other hand, are things like us, organisms that get their food by eating other organisms. So we are autotrophs. The main category of, we are heterotrophs. The main category of autotrophs we tend to hear about are plants. Now, the point of confusion most people have is heterotrophs. Which of these two processes do heterotrophs do? Well, most people accurately say heterotrophs do cellular respiration. So heterotrophs, cellular respiration. We break down the energy of things we eat, and that's absolutely correct. When it comes to autotrophs, the question is, which of these processes do autotrophs do? And most people immediately say they do photosynthesis. I mean, that's kind of the definition. They produce food using photosynthesis. So of course they do photosynthesis. The mistake people make is forgetting that autotrophs also do respiration. Photosynthesis is about creating glucose using sunlight and carbon dioxide and water. But once we have created that glucose, we need to break it down and use it at a later point in time. So while heterotrophs only do cell respiration, autotrophs do both cell respiration and photosynthesis. Okay, when we talk about photosynthesis, and that's going to be our focus today, we need to know where this occurs. Photosynthesis occurs inside the chloroplast, one of the organelles you've been learning about. The chloroplast, here's a diagram, is made up of different parts. You don't need to know all the parts, but you do need to notice a few key features. First off, much like the mitochondria, the chloroplast has a double membrane layer. So we've got multiple membranes, one inside the other. It also has what look like little stacks of discs or pancakes right here. These are called thylakoids. So the individual discs are thylakoids. A stack of them are called the granum. But thylakoid is a term you need to know. It's going to become important. The other characteristic you need to know about is what appears to be the empty space in here. So the space that isn't filled up with anything. Now this is not air. This is called the stroma. And much like the cytoplasm, cytosol, inside a cell, it contains a variety of fluids, jelly-like substances and stuff, and some of our chemical reactions are going to occur in that location. So the chloroplast contains the stroma and stacks of thylakoids that are going to become important in our future conversation. So while the chloroplast is the location of cellular respiration, so while the chloroplast is the location of photosynthesis, what exactly is photosynthesis? Well, photosynthesis can be divided into two different stages. We have the light reaction and the Calvin cycle. The light reaction occurs inside the thylakoid. That's why we need to know those discs inside the chloroplast are called the thylakoids. The light reaction, the first stage of photosynthesis, occurs inside them. And the purpose of the light reaction is to convert light energy into chemical energy. And during the light reaction, we are going to produce ATP and a molecule called NADPH. That might look very similar to what you saw in cellular respiration, NADH, and you're correct. It's an oxidation reduction molecule, and we're going to use it in a very similar way, but it's not the exact same molecule you saw in cell respiration, slightly different, and hence the addition to a P in there. One way to keep them straight is, remember, photosynthesis starts with a P, so the oxidation reduction molecule we're going to use, NADPH, is the one that's used in photosynthesis. NADH is the one used in cellular respiration, which does not start with a P. So that's the light reaction. The second stage is the Calvin cycle. The Calvin cycle is going to occur inside that stroma, the empty space. And the purpose of the Calvin cycle is going to use the ATP and the NADPH from the light reaction and carbon dioxide from the air around the plants. And from those is going to be able to produce carbohydrates, sugar in particular, glucose. Before we start to talk about the details of how this works, though, we need to talk a little bit about light and how light functions and how we're going to use it as part of photosynthesis. Light is something that we see all around us. We see light every time we walk outside, the sun's shining, reflecting off the moon. We see light. When we walk inside, we turn on light switches and we have light all around us. It enables us to see and be active in our environments. But we need to know a few things about light from more of a chemical perspective. The big thing we need to know about light is the fact that light comes in two different forms or two different interpretations of light. We have, first off, particles of light that we call photons. Photons contain energy and are used to power photosynthesis. The other thing is light is often described as wavelengths, these movements of light. We'll talk about wavelengths in just a couple minutes. But with light, one of the things that we're concerned about with light is what happens when light comes down and hits an object, when it encounters an object. So pretend this is an object and I'm shining light on it. Okay, so I'm shining light down toward this object. When it arrives and reaches the object, one of three things is going to happen. The first thing is what we call the light might be transmitted. That means the light's going to come down, it's going to pass through the object and continue on. So that would be our first one, transmitted light. The second thing that might happen is light can come down, hit the object, and bounce off. This would be a case of the light being reflected. And then finally, light can come down and it can hit the object and it can be absorbed by the object. That's the one we're most interested in because that is when it's absorbed, the energy contained in the light is absorbed into the object that it hit. One thing that's kind of interesting about light and the reflected light in particular is this is what determines color. So for example, I have a glass here that's purple in color. The reason it's purple in color is when white light shines on it. Understand the white light contains all the colors. When white light shines and hits that cup, it reflects back only the purple light. All the other colors are absorbed into the cup. Because the purple light is reflected back, it goes to your eye and you see purple light and you interpret that cup as being purple. If we had a yellow cup, the yellow light is being reflected back. So it is possible when white light shines on an object for some of the wavelengths of light to be absorbed, others to be reflected, and still others to be transmitted. So I mentioned wavelengths a minute ago. Let's take a look at wavelengths. Light comes in a wide variety of wavelengths, and wavelengths are generally described at how quickly a wave repeats. Wavelengths on one end of the spectrum can occur very short wavelengths. That means a wavelength goes up and down very, very quickly. Okay, so each wave repeats very, very fast. On the opposite end of the spectrum, we have very long wavelengths, which repeat slowly. These contain different amounts of energy. The very short wavelengths up here at the top contain more energy, and the long wavelengths at the bottom here contain less energy. So different types of light contain different amounts of energy. Now, we tend to think of light as what we call visible light, what we're able to see, but there's actually a much wider range of these wavelengths, and they include many things that we don't even think of as light. For example, the extremely high energy wavelengths, very, very short wavelengths, include cosmic rays and gamma rays. It includes X-rays and ultraviolet light. The longer wavelengths, on the other hand, include infrared and microwaves or radio waves. For photosynthesis, though, we're going to focus on what we call visible light. It's a limited range of wavelengths going from about 400 to 700 nanometers, and these wavelengths are the ones that the human eye is able to see, hence our name, visible light. And different wavelengths are interpreted as different colors. So on our shorter wavelengths, we've got violet, blue, and as we get longer, we move to green, yellow, orange, and finally red. So we have the colors of the rainbow included here. In photosynthesis, visible light will hit a plant and is going to be able to absorb some wavelengths, and it will reflect other wavelengths. And different wavelengths will provide different amounts of energy to be used to power the process of photosynthesis. So how do we actually absorb wavelengths? Well, generally to do that, we're going to use what are called pigments. Pigments are molecules that absorb various wavelengths. So pigments absorb wavelengths of light, or absorb light. And there are a variety of different types of pigments out there, but the main one that we use in photosynthesis is called chlorophyll. Chlorophyll is by far the most abundant type of pigment used in photosynthesis. And this graph here represents what we call an absorption spectrum. It is telling us how well certain pigments absorb different wavelengths of light. And chlorophyll is represented by the green graph here. If you look at this closely, what you'll see is that chlorophyll absorbs light very, very well in the blue to purple range, and also very, very well in that deep red range. But it's not very good at absorbing light in the greens, yellows. It gets a little better as we start going into oranges. But by far the best absorption for chlorophyll is in this blue, purple, and red range. One indication that this is true is by taking a look at the color of plants. So look at our picture of a plant here. What color do you see? Green. That's because chlorophyll reflects green light. It does not absorb it. Therefore, it makes sense that it is terrible at using green light in order for photosynthesis to occur. But what about the other colors of light? Are we able to use them at all? Fortunately, we are, because the plant cells, in addition to chlorophyll, have other pigments. And they have a series of pigments called carotenoids. Carotenoids are a series of other pigments that have somewhat different absorption spectrums to chlorophyll. This blue line here represents one carotenoid. You'll see, much like chlorophyll, it's very good at absorbing that violet and blue light as well as red light. Not quite as good as chlorophyll, but it absorbs them pretty well. However, it is much better at absorbing the greens, yellows, and oranges. So inside any given plant, it's going to have a combination of chlorophyll, the majority of the pigment, but also carotenoids that are in there able to absorb some of the other spectrum, trying to maximize the amount of energy a plant's able to use for photosynthesis. You've actually seen carotenoids, whether you know it or not. In the fall, many plants and trees begin to lose their leaves. And when they do that, the pigments begin to break down. Well, chlorophyll breaks down much more rapidly than carotenoids do. And this is why in the fall, green leaves turn orange, yellow, and red. You're seeing the chlorophyll disappear and the carotenoids are revealed until the carotenoids break down, and then the leaves lose all color. So we've got pigments. We know they absorb energy, and we're going to use that. But how exactly do they absorb that energy? To understand that, we're going to take our next step and look at what we call a photosystem. This is a photosystem. Photosystems occur in the thylakoid membrane. So this is our membrane. This is outside the thylakoid, but inside the chloroplast, and this is inside. A photosystem is composed of numerous protein molecules, and it also contains a number of pigment molecules. So looking at this, each of these right here are a pigment molecule. Most of the time chlorophyll, though some of them might be carotenoids. What is going to happen is a photon of light, wavelength, is going to come down here and it's going to land on one of our pigment molecules and it's going to be absorbed. So the energy from that photon has now been absorbed. It could land on any of these chlorophyll molecules, it doesn't matter, but it lands on one of them. However, what I want to do is the photosystem wants to take all the energy and bring it down here into one location, what we call the reaction center. So after the photon is absorbed by this chlorophyll molecule, it's going to pass that energy to this chlorophyll molecule, which is going to pass it to this chlorophyll molecule. And finally, it's going to pass it over to our reaction center. This is going to happen multiple times. Another photon is going to come down, hit this one, get passed over, get passed over, get passed over. What's happening in the reaction center is in the reaction center we have an electron. That electron is going to get excited. Okay, it's going to get energized. It's going to absorb energy. And once it reaches an excited state, being represented by this arrow, making it more and more excited, once it reaches this excited state, it is going to get passed on to the beginning of an electron transport chain that we'll talk about in a moment. So this excited electron gets passed over to an electron transport chain. The photosystem's job is to absorb photons, pass the energy absorbed to the reaction center, and that energy is then used to excite an electron. Let's talk for a moment exactly what we mean when we say excite an electron. If you remember back to chemistry, you have molecules, for example, hydrogen. And hydrogen will have an orbital that contains an electron molecule. I'm sorry, it's not a molecule, but we have an electron on here. Now, as molecules contain more and more electrons, they have different layers of orbitals. So here's another orbital layer, and there's more and more. Electrons fill the smallest, the lowest orbital first, and then once that's filled, they'll move to the next one and the next one and the next one, depending on how many electrons they have. When I say that the electron is getting excited, what that means is this electron has gained enough energy that it is going to jump to the next orbital. It is now in this next orbital. It has so much energy. It is highly reactive. It wants to react with something. And until it does react or uses up that energy, it'll stay in that heightened orbital. And once the energy is gone, it will return to its normal orbital. So when I say this electron is excited, it contains a lot of energy and it's highly reactive. It wants to react to things. And we're going to use that to power this process in just a couple minutes. Okay, we have set the stage. Now it's time to look at the light reaction and understand what's going on. So this is an example of the light reaction. This is our first of two stages for photosynthesis. What I would like you to do for a moment is, in just a second, I want you to pause the video. I want you to look at this a little and see, do you see any similarities between this diagram and what you learned about in cellular respiration? There's several of them, so I want to see if you can start making connections between the two now before I explain them. So go ahead, stop the video, and once you think you've identified them, go ahead and start running again, and we'll go through it. Okay, how'd you do? Did you find any similarities? Okay, I'm going to point out three particular similarities. The first thing is we have an electron transport chain that is moving hydrogen from outside to inside the membrane. So we saw that in the electron transport chain of cellular respiration. Secondly, I have an oxidation reduction reaction happening here. True, it's a different molecule. It's NADP plus becoming NADPH instead of NAD plus becoming NADH, but it's still a oxidation reduction reaction. And finally, I have my ATP synthase, the exact same molecule I used to create ATP in the electron transport chain, is creating ATP here. Okay, so let's take a look at the details of this whole process and see how this works. In the light reaction, we are working in the thylakoid membrane. So you can see right here, we've got a thylakoid and we're in the membrane. So this is outside the membrane in the stroma, and this is inside the membrane, inside the actual thylakoid. Embedded in this membrane are two photosystems. One of them is called photosystem number 2. The other is photosystem number 1. And one point of confusion often is the order they occur. Photosystem 2 occurs first, photosystem 1 occurs second. I sometimes feel this was done just to confuse everyone, but in reality it was done because photosystem 1 was discovered first, photosystem 2 was discovered later, and that's where the naming came from. Okay, so don't get confused by it. Photosystem 2 does its job first, then photosystem 1 will do its job. We also have a series of proteins that are embedded in the membrane, forming electron transport chains. So let's take a look at what happens. My first photosystem, we already talked about what it does. It captures photons of light and it uses them to energize electrons. That's what this photosystem is doing. Where does the electron come from? The electron comes from water. We have a water molecule that is going to be broken apart. The electron that is broken off is going to be handed off to the photosystem, and that is the electron that is getting energized. The remainder, the oxygen, is a waste product. Remember, one of our waste products for photosynthesis is oxygen. And hydrogen ions are going to be released here inside the thylakoid to help build a concentration gradient. You might notice that we have half an oxygen. There's no such thing as half an oxygen. They're just trying to keep it simple. They're not balancing a chemical equation like you normally would. Okay, so understand, oxygen's being released. It's not half. Two water molecules go through this process at the same time. Okay, so we have an electron now that has been energized by photosystem 2. That electron is going to be passed down an electron transport chain, moving from one protein to the next. During this process, the energy from the electron is going to pull one hydrogen proton across the membrane from the stroma into the thylakoid, helping to create a concentration gradient of hydrogen inside the thylakoid. After that, the electron has used up its energy, so it's going to be passed on to another photosystem, photosystem 1. This photosystem works the exact same way. You might notice the P700 here and the P680 in the first one. Those are just official naming for it. Don't worry about them. Act like those numbers don't exist. So the electron, the same electron from this water molecule, not a new electron, the same one, is going to get energized again. New light comes down, is absorbed, energizes our electron, and that electron is passed over on another electron transport chain. But this one's a little different. This electron transport chain does not move any hydrogens. That step is done. Instead, after being passed off a couple times, this electron undergoes an oxidation reduction reaction with NADP+. NADP+ is going to be reduced. It's going to bond with NADP+ is going to bond with the electron and hydrogen to become NADPH. NADPH is then going to go to the Calvin cycle. So we're going to use it in a few minutes, but not yet. So NADP+ is reduced. This protein is oxidized, and so that's our oxidation reduction reaction. The last step here is we are going to use this concentration gradient that we created with the electrons going through an ATP synthase, which works the exact same way it does in the electron transport chain. It's going to open up. The hydrogen is going to move through it because of the concentration gradient. It's going to move out here. And as it moves, it causes the ATP synthase to spin around. And that kinetic energy is going to be used to make ADP into ATP. This ATP molecule is also going to go to the Calvin cycle. Now you might be looking at this and wondering, like, wait a minute, if we can make ATP, why on earth are we doing photosynthesis? Why not just use that ATP? We could just take away cell respiration. And well, that might make sense. It doesn't really work that way. This doesn't produce as much ATP, and we are actually going to use the ATP in the Calvin cycle. So we need another source of ATP, which is cellular respiration. Okay, hopefully you understand the light reaction now and how it's used to create NADPH and ATP molecules, both of which are going to go to the Calvin cycle. And they're going to power the Calvin cycle. So with that in mind, let's go look at the Calvin cycle. The Calvin cycle probably reminds you a little bit of the Krebs cycle. We've got a series of chemical reactions here, and it's in a giant circle. Because it's in a circle, you can technically start anywhere you want, but I'm going to start over here. That's going to be my beginning point. The Calvin cycle is broken up into three big steps. The first step is carbon fixation, then the reduction in sugar production, and finally the regeneration of RUBP. So let's talk about this and see how this works. We're going to start off with 6 RUBP molecules. RUBP is an abbreviation of a very long name. You don't need to have the name memorized, but if you're ever told RUBP or see the name and then RUBP in parentheses, this is the molecule they're talking about. Much like the diagrams we use for respiration, this one is showing you the carbons. It also is including phosphates, but understand there are hydrogens, oxygens, and other things bonded onto this molecule. What we want to do here is we are going to take RUBP and we are going to combine it with 6 carbon dioxide molecules. The carbon dioxide comes from outside the plant, just in the air. So it is absorbing carbon dioxide here. And we have RUBP, which is 5 carbons long. So 1, 2, 3, 4, 5. And we're going to add a carbon dioxide. We're going to add this series of reactions. What's going to happen is we're going to add a 6th carbon to it. And immediately after doing that, we're going to break this molecule in half. So we have 3 carbons, 3 carbons. So we started off with 6 5-carbon molecules. We added a carbon dioxide, a carbon to each of those. So now we have 6 6-carbon molecules. And we break them all in half. So we end up with 12 3-carbon molecules. These new molecules are called 3PG. The 3 has to do with where the phosphate is bonded to. And you can see they have 3 carbons, half of what the molecule we just broke in half, and that's why we have 12 of them. That's carbon fixation. The purpose of carbon fixation is simply to bring extra carbon in and bond it into these molecules. And the source of that carbon is carbon dioxide. The one other thing you need to know about carbon fixation is the enzyme used in the process. The enzyme is called rubisco. So rubisco. Rubisco is the most abundant protein on the planet. It is the enzyme that drives carbon fixation. It bonds the carbon dioxide to the RUBP. It runs a very, very important job. And as I said, it's the most common protein on the planet. The reason for that is partly as a very important job, but also it's an incredibly slow enzyme. Rubisco can only do 2 to 3 carbon fixations a minute. Per second. But considering the amount of photosynthesis that happens in the world, that's not very fast. The other thing that we'll talk about later on is rubisco is a fairly clumsy enzyme. It messes up a lot. And so that's why the sheer abundance of rubisco out there. Rubisco is, I believe, the only enzyme in this course you will ever hear about that does not end in ACE. So know it's an enzyme, understand it's used for carbon fixation. We now go into reduction and sugar production. We have 12 3PG molecules. Our goal is to change these 3PG molecules into 12 G3P molecules. I know they look the same. They look like they both have 3 carbons and a phosphate, and they do. But there are a variety of chemical differences. They are not the same molecule. We're going to change them from 3PG into G3P. How are we going to do that? Well, we're going to provide energy in 2 forms. The first part of the energy is going to be ATP. 12 ATP that came from the light reaction are going to be used. They become ADP. And as a result, we're going to start changing the 3PG molecules. The second thing that's going to happen is the NADPH also came from the light reaction. We're going to use those. The NADPH is going to be oxidized. It's now NADP+. Again, it's going to go back to the light reaction. It can be used again. But the electrons are going to be passed over to this unnamed molecule, providing energy to it. And in the end, we're going to end up with 12 G3P molecules that have a different energy profile. They actually contain more energy than our original 3PG molecules did. So the light reaction provided the energy in the form of ATP and NADPH to make 3PG into G3P molecules. We are now ready to make our sugars. We have 12 3PG molecules. 2 of them are going to break off, and they're going to go down, and they are going to be transformed into glucose, into a sugar molecule. Okay. How are they going to do that? It's a series of reactions. You do not need to know the details, but understand 2 of these molecules are going to go off, and they are going to become sugar. We now are left with 10 G3P molecules, and our goal is to turn these 10 3PG molecules back into the 6 RUBP molecules that we started with. That is going to take a series of chemical reactions, and it does not happen naturally, so it's going to require an energy source. The energy is provided by ATP, which once again came from the light reaction. This ATP is going to become ADP, and these 10 G3Ps are going to be modified, broken apart, reassembled until they form 6 RUBP molecules. We're now back at the beginning of our cycle, and we're able to repeat that cycle over and over and over again. I want to point out, why do we need the input of carbon dioxide every time? 6 carbon dioxides. We need it because every cycle we're losing carbon to become sugar. So we need to replace that carbon, and carbon dioxide is the source for providing that extra carbon so we can keep this cycle going forever without running out of carbon. So that's the carbon cycle. Hopefully that made sense. Okay? But I need to point out a possible point of confusion that you might have picked up on or you might not have. If you take a look at the Calvin cycle as I have it set up here, how many ATP molecules do I use? And how many NADPH do I use? Well, if you count carefully, I've got 12 NADPH molecules, but I have 18 ATP molecules. How on earth does the light reaction produce more ATP than NADPH? I don't want to produce extra NADPH because I'm wasting energy when I do that, but I have to produce enough ATP in order to be able to power the entire chemical reaction. So the question is, how do I actually balance that? To answer that question, we're going to have to go back to the light reaction and add a little detail that I left out the first time. So let's do that. So here we are back at the light reaction. The light reaction actually has two different versions. The one I showed you before is what we call the non-cyclic electron flow, and this is the traditional method. The non-cyclic electron flow follows exactly what I told you before. The electron is energized, it goes down an electron transport chain, which brings one hydrogen across. That hydrogen produces one ATP. This electron is powered again. It goes here, and it is used to reduce one NADP plus to NADPH. So the non-cyclic electron flow produces one ATP and one NADPH for each electron that passes through. That's the normal. That would give us, for 12 electrons going through, 12 ATP, 12 NADPH. But as we saw a minute ago, I actually need 18 ATP. So how do I fix this discrepancy? The solution is to use what is called the cyclic electron flow. 1 out of every 2 electrons follow an alternate pathway. So half of the electrons going through are going to follow the non-cyclic electron flow, exactly what I showed you above. This one, however, is going to do something slightly different. The electron is going to get energized, just like normal. It's going to go down an electron transport chain, pulling a hydrogen through. But when it reaches the second photosystem, photosystem 1, it is going to get energized again, but instead of being passed on and be joining with NADP+, it is going to be passed back through the original electron transport chain one more time, and then back to the photosystem. The second time it enters the photosystem, it will go up, energized, and be passed over and join with NADP+ to become NADPH. Okay, so once again, the electron will be energized. It will go through the electron transport chain, pulling a hydrogen. It gets energized and it does it again, pulling a second hydrogen. And then it gets energized and goes over and gets reduced. So let's look on our diagram here and see exactly what effect that will have. Following the cyclic method, our electron will come through, be energized, pull an ATP, or hydrogen through, which is going to create an ATP molecule. It will get energized, but it will be passed through this one more time, pulling a second hydrogen through, which is going to produce another ATP molecule. The electron will then be energized and passed over here and be joined for a reduction of NADP+ to NADPH. Because half of the electrons, so 6 electrons, will go through the non-cyclic. 6 of them will produce 1 ATP and 1 NADPH. The other 6 electrons are going to go through the cyclic version, where they each produce 2 ATP molecules and 1 NADPH molecule. At the end of this, we end up with a total of 18 ATP and 12 NADPH molecules. So that's how your cells have solved this problem. That is photosynthesis. My hope is it makes sense, you understand it. I encourage you to look for similarities between photosynthesis and cellular respiration. They share a lot of them, and if you can understand the processes and just fit them in the right place, it's much easier rather than just trying to memorize 2 systems. Much like respiration, you don't have to memorize every step. What you need to know the overall process, its purpose, what it does, the main purpose of each step, the light reaction, as well as the Calvin cycle. You need to know the inputs and outputs. You need to know where each occurs. And you need to be able to look at and interpret diagrams of varying complexity. I encourage you to practice drawing it to gain a better understanding. Draw it and explain it. If you keep that up, I guarantee photosynthesis is going to make a lot of sense to you. Okay, thank you very much. I hope you got something out of this. And if you have questions, make sure to bring them into class.